in our series at Medical Sciences. Today we want to take you through a set of 4 biochemical tests that are performed to identify gram-negative bacteria, what we call enterobacteriation. And these tests are Indole, Methyl red, Voges-Proskauer, and Citrate utilization tests. And if you're new on this channel, please, you're reminded to subscribe as a way of supporting us. Like the video and share to many others. So want to see these 4 tests, which is a set of 4 tests. How are they utilized in identification of enterobacteriation or enteric bacilli? And we have said I stands for Indole, whereas M is for methyl red, VI is for VP, that is the Voges-Proskauer, where I does not indicate anything but is usually just to bring the mnemonic. So here the most important thing which is important is the V, which stands for Voges-Proskauer. Then C is for citrate utilization tests. So in this video we are going to see the principles, briefly about the principles of these tests and which organisms do they identify when they are positive. And without wasting time, we want to begin with Indole. Indole test, this one utilizes the enzyme that if we have a protein tryptophan, this tryptophan is converted to indole by an enzyme known as tryptophanase. And on addition of Kovacs reagent, this indole forms a red ring on the surface of the broth. So you'll find if you have the broth containing indicator, which is methyl red, phenol red indicator, so we shall form what we call a red ring on the surface on addition of Kovacs reagent. And the positive organisms for this is E. coli and Proteus vulgaris. These are some of the peculiar examples which are positive with indole, that they are able, this E. coli is able to produce enzyme tryptophanase that utilizes tryptophan in the broth to produce indole, which reacts with the Kovacs reagent to produce a red colored ring on the surface of the broth. So we are utilizing indole to confirm E. coli and Proteus vulgaris. Then this was number 1. Number 2 we can talk about is methyl red test. And this, it is abbreviated as MR. Methyl red, it means this identification biochemical test identifies using indicator methyl red. So this one detects if you have a tube, you add what we call glucose phosphate broth. A phosphate is acting as a buffer to maintain neutral pH. So this broth, if I come and I add an organism which is able to ferment glucose, it is going to ferment glucose to produce the acid. That is pyruvic acid. And this acid will return the indicator, methyl red indicator, and it will turn to red color. It will give a positive result, gives a red color, meaning that this organism is able to ferment glucose, to produce pyruvic acid, which turns the indicator from yellow to red. So initially the indicator was yellow, but when we produce the acid, we form a red color. And examples of organisms which are positive with methyl red test are the same. That is E. coli and Proteus vulgaris. They also give positive results with the methyl red test. Maybe another one I can add is Salmonella. Apart from this, also Salmonella. So we have E. coli, we have Proteus vulgaris, we have Salmonella, which can also show, and even Shigella species. They show positive with the methyl red test, meaning they are able to utilize glucose only to produce the acid that turns the indicator. Then the third test we can utilize in our video is the V, which is Voges-Proskauer, which we abbreviate as VP. This test, it looks if I have a broth and the broth still utilizes the same component, that is glucose, phosphate broth at neutral pH. As we said, the phosphate is acting as the buffer. So when we come and add the organism, we are which is in question, that is the enterobacteriation. We are seeing how it is able to ferment the glucose in the broth to produce the pyruvic acid. And if it produces, but this one is looking at the production of the pyruvic acid, which metabolizes further to form what we call acetone. The name of this acetone, you can call it acetyl methylcarbinol. Acetyl methylcarbinol is the chemical name for the acetone which is formed when the organism is able to ferment the glucose. Then after forming acetone, we come and add two reagents, that is 40%. We add 40% potassium hydroxide plus 5% naphthalene. So 40% potassium hydroxide and 5% naphthalene are added, leading to production of red color. So red color is formed due to the formation of diacetyl. So this one is due to the formation of diacetyl compound on addition of 40% potassium hydroxide and 5% alpha naphthalene, which converts acetone to diacetyl compound, which produces the red color. And which organisms are positive with Voges-Proskauer? Here, the organisms that are positive with this test include Voges-Proskauer. We see Enterobacter. Organisms which are positive include Enterobacter aerogenes, Enterobacter species. We are going to see organisms like Klebsiella pneumoniae, produce a positive result with Voges-Proskauer. And another organisms which can produce is Serratia marcescens, also is able to produce a positive result with the VP tests. So this is what we can see under VP organisms: Enterobacter species, Klebsiella pneumoniae, and Serratia, which are able to ferment the glucose, produce acetone compound, which on addition of 40% potassium hydroxide and naphtha form a diacetyl, indicating a positive test. Then the lastly among the four is the citrate. Citrate utilization test. This citrate utilization test is looking for the organisms. It is looking for the organisms which are able to utilize citrate as the sole source of carbon plus energy. So these organisms use only citrate, utilize only citrate as their source of energy and carbon. So what we use is we use Simon's citrate agar. This Simon's citrate agar contains sodium citrate, and this one is always green in color. This broth is always green in color. But when we come and we inoculate the organism, which we suspect to utilize only citrate, is able to use citrate as a source of carbon and energy, and this one leads to formation of the bicarbonate, which turns the pH to alkaline, leading to formation of blue color. So the blue color formed during citrate utilization test indicates a positive result. It means this organism was able to break down the sodium citrate, using it as a source of carbon and energy, hence producing the bicarbonate that produces the alkaline pH, turning the indicator from green to blue. And this one indicates a positive result. Which organisms are able to do this? Organisms include Pseudomonas aeruginosa. Aeruginosa can do that. We can see organisms like Salmonella. Salmonella species also can utilize citrate as a source of carbon, not forgetting Shigella. So organisms here which are citrate utilization positive, which produce the blue color, is Pseudomonas, Salmonella, and Shigella. So if you have your broth, it was green, but at the end of the reaction, it turns blue color. This one indicates a positive test with either Pseudomonas, Salmonella, and Shigella. So after looking at the individual test, now the next step we are going to see is how can we interpret these results. I have here I have M, that is methyl red, I have V, then I have C. So if I have these ones, and you find the organism is positive for Indole, it is positive for methyl red, negative for VP, and negative for citrate utilization agar, here we can suspect organisms like E. coli and Proteus vulgaris. These are the two organisms which we can isolate when they give positive Indole, positive methyl red, then negative. One thing I have not told you is that methyl red and VP, they are alternate, or they are opposite. They give opposite reaction. If an organism is methyl red positive, automatically it is VP negative. And if an organism is negative for methyl red, it is positive. It can be positive for VP. So that is what I want you also to note about these two tests. So it is hard to find an organism which is both positive for methyl red and VP. Either it is positive for methyl red, negative for VP. Then in case we get results whereby here negative Indole, negative methyl red, but positive VP and positive citrate utilization. Here we have organisms like Enterobacter species. We have organisms like Klebsiella pneumoniae, and we can also see organisms like Serratia marcescens. So these are some of the organisms which give negative Indole, positive methyl red, then positive VP and positive citrate. Then which organisms can give, you find an organism giving a negative Indole, a positive methyl red, a negative VP, and a positive citrate utilization. As we said, organisms cannot be both methyl and VP positive. Either it is one of them. So an example of an organism which is positive for methyl red and positive for citrate is Salmonella. Salmonella species, also Shigella. So these ones, they can be positive for methyl red and citrate utilization. Then the last scenario is whereby you find an organism is only positive for citrate. You find it is negative for Indole, negative for methyl red, negative for VP, but positive for citrate. And here a peculiar example is Pseudomonas aeruginosa is positive only for citrate utilization tests. So this is how we can be able to interpret our IMViC biochemical test, which is a set of 4 tests being utilized to identify the family of Enterobacteriaceae. Those are gram-negative bacteria. Hope it has been so helpful to you. Thank you so much for learning with us at Medical Sciences by Naphtali Muhumuza.